Today, I'll be talking on the module Human Chromosome Complements. The basic objective of this lecture is to make you all understand the structure of chromosome, the number of chromosome present in our human body cell. Well, chromosome is derived from two words. Chroma, which means color, and somes means body. So, chromosomes means simply colored thread-like or filamentous body, which are present in the nucleoplasm of living cells. That means in the nucleus of living cells. Basically, they are carrier of genes, and genes are unit of heredity. That means they help in inheritance or transmission of characters from one generation to the next. Now, these chromosomes were first observed in plant cells by Carl Wilhelm von Nageli in 1842, and their behavior was described first by Walter Fleming in 1882. And this particular term, chromosome, was coined by another German anatomist, von Weyl in 1888. Now, coming to the makeup of chromosome. Chromosome is made up of proteins and nucleic acid. That is DNA, deoxyribonucleic acid. And structurally, chromosome is made up of two chromatids. That means two arms, double-stranded DNA, and protein. And these two double strands are bound together at the primary constriction, which is called a centromere. Therefore, a centromere usually divides the chromosome into two arms. One of the arm is comparatively short, another arm may be comparatively long, or maybe equal. But if one arm is short and the other arm is long, the shorter arm is called P arm and the longer arm is called Q arm. And in a chromosome, the P arms always are arranged in a top orientation, in a top position, whereas the Q arms face downward. And on the basis of the position of centromere, we can classify chromosomes into four types. One is metacentric. So metacentric chromosomes are those chromosomes in which the centromere is median or nearly in the middle of the arms. And such chromosome therefore has two well-defined arms with a length ratio varying from 1:1 to 2.5:1. Then we have the second category of or second type of chromosome, that is submetacentric chromosomes. They have unequal arms and is almost L-shaped. The ratio of the arms lie in between the ratio of metacentric chromosome and acrocentric chromosome. Now, let us come to the third type of chromosome, that is acrocentric chromosome. Here, in acrocentric chromosome, centromere is close to one end of the chromosome. As such, one arm is substantially smaller than the other. And the ratio ranges from 3:1 to 10:1. Then finally we have 4 types of chromosome, that is telocentric chromosome. Here in telocentric chromosome, centromere is strictly terminal. That means chromosome has only one arm, the terminal part being the centromere. So these are different types of chromosome based on the position of centromere. Now coming to the karyotyping, that means chromosomal complement or chromosome complement. A karyotype is derived from a Greek word karyon, which means kernel, seed or nucleus. So karyotype is therefore the number and appearance of chromosome in the nucleus of a eukaryotic cell. The term karyotype is also used for the complete set of chromosome or chromosome complement in a species or an individual organism. Well, karyotype therefore refers to the complement of chromosomes either at species level or of individuals. It describes the number of chromosomes structure under the light microscope, and while doing so, due importance should be given to their length, that means length of the arms, the position of the centromere, the bending pattern, any other difference between sex chromosome, and also any other physical characteristics. So therefore, when we talk of chromosome complement, we look into various characteristics of a chromosome for identifying, for systematically grouping chromosomes. The technique of determining the karyotype is usually called karyotyping. And the study of whole sets of chromosomes is sometimes known as karyology. And when chromosomes are arranged in a standard format, that means in pairs, because chromosome usually occurs in pairs. So when they are arranged in standard format in pairs and according to the size and position of centromere, then it is known as a karyogram or a diagram. Let us go back little to the historical perspective of human karyotyping. It was in 1912 that Hans von Vidya-mitra reported that there are 47 chromosomes in spermatogonia and 48 chromosomes in oogonia, thereby concluding that females have XX chromosome and males have X0 or XO chromosome. This was the sex determining mechanism explained by Vidya-mitra. Well, later on, it was Spencer in the year 1922, though he was not certain whether the diploid of human was 46 or 48. He was first favoring for 46, but later on revised his opinion from 46 to 48. And he correctly, however, insisted on human having an XX or XY system. So number of human chromosome remain at 48 chromosomes for over nearly 30 years. And finally, it was Jo-hin Jio on the basis of his publication in 1956 that the normal human chromosome complement or normal human karyotype comprises of 23 pairs of chromosome, thereby making 46 in total. Now, we have well known and it is already established that human beings have 46 chromosomes or 23 pairs of chromosome. Well, coming to the human somatic or body cells, it has two complete sets of chromosome given by each parent, and these two sets together constitute the diploid condition represented by 2n. That means one n given by mother, another n given by father, constituting twice n in diploid condition. Therefore, this twice n chromosome of human is 46 in number. Coming to the sex cell, that is the gametes, that means eggs and the sperm, have only one set, that is 23 chromosomes, and it is known as a haploid condition. So here it means we have two types of chromosome: autosomes having 22 pairs or 44 chromosomes, and gonosomes or sex chromosome with one pair or two chromosomes. The sex chromosome of male is XY, that is hemizygous, and that of female is represented by XX, homozygous. The human normal karyotype, when observed under microscope, particularly at the mitotic metaphase stage, we can see 46 chromosome, meaning that 46 colored blobs, two of which are either XX or XY. Now let us go a little bit deeper on human chromosome complement. These 46 chromosomes can be divided into 7 different groups on the basis of the structure of the chromosome, on the basis of length of the arm from the centromere. Now, these 7 groups, in the first group, that is group A, the chromosome number 1, 2 and 3, they are kept under this group 1 or group A, the first group I mean. So these chromosome number 1, 2 and 3 are large metracentric chromosomes. And in group number B, the chromosome number 4 and 5 comes. And they are large submetacentric and are all similar. And in the third group, that is group C, the chromosome number 6, 7, 8, 9, 10, 11 and 12, that is from chromosome number 6 to 12, along with X chromosome, are kept in this group. Structurally, they are submetacentric and of medium sized. The next one is the fourth group. Under this fourth group, chromosome number 13, 14 and 15 comes. They are again medium sized but acrocentric. That means having a very short arm and a very comparatively long arm, plus a satellite. So they are medium sized. Coming to the fifth group, that is group E, chromosome number 16, 17 and 18 comes under this group. They are short in length, they are metacentric. However, the chromosome number 17, 18 are submetacentric. That means out of these three, the 16th chromosome is short and metacentric, and the chromosome number 17 and 18 are submetacentric. Coming to the sixth group, that is F group, we have chromosome number 19 and 20. They are again short and metacentric chromosomes. In the last group, that is group Z, we have chromosome number 21, 22 and Y. They are basically short acrocentric with satellite. However, in case of Y, there is no satellite. So these are different types of chromosome or different groups of chromosome of human body. So when we talk of chromosome complement, we mean these 7 different groups. But these 7 different groups are basically the normal human chromosome complement. Now, apart from this, we have other abnormal human chromosome complements. One such condition is Down syndrome, which we often call Mongoloid syndrome. Here, there is one extra copy of 21st chromosome. So that is why we call it as trisomy chromosome. So therefore, in a normal human karyotype, we have 46 chromosomes. But in case of Down syndrome, we have 47 chromosomes. And the extra chromosome is a copy of 21st chromosome. Then we have another example, that is Edwards syndrome. An individual with Edwards syndrome will have 47 chromosomes. That means 46 plus one extra copy of 18th chromosome. So usually individual with such 18th extra chromosome have retardation of motor functioning, a number of congenital diseases or abnormalities causing serious health problems, and usually 90% of those affected by this Edwards syndrome die in infancy. Now, another third category is Klinefelter's syndrome. Here, the chromosome complement is XXY. So when it is XX, it is female. When it is XY, it is male. So whether one is female or male is determined by Y. So here it is having both the characteristics of female as well as male. So but basically Klinefelter syndrome individuals are men and are usually sterile, and they tend to be taller and heavier, have longer arms and legs, and boys with such a syndrome are often shy and quiet and have higher incidence of speech delay and dyslexia. And without testosterone treatment, some may develop gynecomastia during puberty. When again we have another syndrome, that is Patau syndrome. This is called D syndrome or trisomy 13. That means extra copy of 13 chromosome. Then we have similarly triple X syndrome having XXX. So here XX simply denotes female, but again another X chromosome, extra X chromosome. So triple X syndrome. So such girls tends to be tall and thin and have higher incidence of dyslexia. Then there is a very popular syndrome known as Turner syndrome. Now in Turner syndrome, there is only one X instead of XX or XY. That means either X or Y is missing. So there will be only one X chromosome. So then naturally the chromosome complement of such individual will be X0 chromosome. Therefore, Turner syndrome or individuals with Turner syndrome often have a short stature, low hairline, abnormal eye feature and bone development, and a caved-in appearance to the chest. Similarly, we have XYY syndrome. Here, XYY definitely should be a boy because XY denotes boy. And there is another addition of Y chromosome. So chromosome complement will be 47, 46 plus an extra Y chromosome. So such boys are usually taller than their siblings. Like XXYs, boys and the girls, like XXY boys and XXX girls, they are more likely to have learning difficulties. In short, we have learned that human beings have 46 chromosomes, which include XX in case of female and XY in case of males. And there are also cases of abnormal human karyotype, which can be exemplified by a number of chromosome anomalies, abnormal chromosome complements like Down syndrome, Edwards syndrome, Patau syndrome, Turner syndromes, Klinefelter syndrome, etc. Thank you.